So first I would like to thank the organizing committee and Victor if it's here, I don't know, and for inviting me and being with you here. And it's a great opportunity for me to again learn what happened since the last few years in this topic. And actually many of the knowledge that I will present to you are already published, like you guys, like the scientists here, many of them, so I'm going to cite their works on this field, but I especially going to be interested in aging part of deuterium. So my main field of research is on aging, and my interest to deuterium also started with concerning its relation that I thought that those times with aging. So this is how I will present my talk here. First, I make some historical perspectives, and I give some critical basic info that you already know and presented too many times here. And then I'll talk about the role of CD bond. It's a very strong bond, to be maybe not easy to break it, and therefore maybe the accumulation of NADD in cells. And then I'll talk about proton channels, because my main work on this field is about 80% is rotor part, stuttering of rotor part by the entrance of a deuteron, and then stuttering 80% is, and then H2O reactome. I'll talk about it because there are up to maybe more than 4,000 different reactions in KEGG pathway that use water as an input or output. So I think we need to look all of them separately. And I'll mention some of the especially historical studies related to aging and cancer, and related studies related to heavy water. And I'll talk about lysosomal pH change, because lysosomal pH was shown was one of the very earliest marker for cellular senescence. So therefore, I'm going to again suggest VATPases, like normal ATPase in mitochondria, is one of the targets for detonation and maybe stuttering of VATPases. So when we look at the PubMed, so one of the earliest, the first article that comes is from Balinkin, and he mentions Professor Levi's studies. And Professor Levi's tested the effect of 100% heavy water in the sprouting of the tobacco seeds, and it inhibited completely. And then he's a very interesting suggestion from Balinkin. He says that there should be a kind of mechanism in cells or tissues that selects for deuterium, and therefore we would expect high concentration of deuterium outside the cell but low concentration inside the cell. I think it's very interesting, but this is just maybe 1933s. And he says, therefore, that if heavy water is toxic, then we would expect a kind of beneficial effect from light water. And when we look at the Lewis studies, so it's published in this year, it's one of the, I think, the first article in biology for related, maybe if you know, one of the earliest ones, but as I found, so this is the first one. It's published related to this field on the biological effects of deuterium. So it showed the toxicity and lethal doses in many different systems, biologicals. He also wanted to test its effect in mice, but during those times, he says that it was very expensive to produce light water. Therefore, he couldn't reach the amount that can be used for mice. Therefore, he couldn't finish his studies in mice. But later on, after 50s, so it was shown that
and the heavy water
is toxic and even lethal for animals. And I think one of the other pioneers in the field is Urey, and he says that I think this word is very, very important from pragmatically especially. He says that the differences in physical and chemical properties of compounds of hydrogen and deuterium are much greater than in the case of any other two isotopes which have been investigated thus far. So this means, and I believe in that way also, one of the main barriers in our minds as scientists is accepting deuterium only as an isotope yes, true, it's an isotope, but pragmatically, I think we should accept deuterium as a kind of different entity, as a kind of a specific element in order to prevent those kind, because since its abundance is too much compared to other isotopes, so there should be some systems, enzymes, proteins, or other macromolecules that selectively use deuterium instead of hydrogen. So these are general info about the abundance of deuterium in nature, and it will look, especially another scientist also mentioned here, the viscosity of deuterium is up to 25% higher, and this is one of the main reasons maybe for some effects of heavy water. So when we look at the hydrogen, it makes up to 10% of human body. So for every 6,600 approximate hydrogens, proteins, we have one deuterium. But in water, the amount goes to 1 to 3,000 because it contains two hydrogens. And when we look, as many other scientists mentioned here, it's, for example, when we look at potassium, its concentrations almost quadruples the amount of potassium in body fluids. But almost everyone, maybe except the audience here, is almost ignorant about the presence of high concentrations of deuterium in biological fluids, in our body fluids. And it's neglected. So one of the greatest examples for related, especially longevity and health span, or maybe in the field of cancer also, or caloric restriction, it comes from NADH. One of the modern or nowadays NAD plus concentrations, or maintaining NAD levels in cells, is accepted as a very critical component, especially because of the sirtuins. Sirtuins are NAD dependent histone deacetylase proteins, and they have a huge effect on maintaining or increasing lifespan or age-related damage repairing systems. Therefore, sirtuins require NAD, but once NAD plus is bound with deuterium instead of proton or hydrogen, so it's going to be slower to break NAD bond. Therefore, it can be expected that the amount of steady state level, amount of NADD can increase during passage of time, and this can be one of the barriers to maintaining the NAD pool that is required for the assurance of longevity mechanisms. Therefore, maybe it would be interesting to check the levels, or if there's an accumulation of NAD levels in aging cells or organisms. So one of the other mechanisms to maintain NAD plus levels in the cells is through, especially in cancer cells, is related to lactic acid's conversion to pyruvate. By lactate dehydrogenase, it uses NADH to produce NAD plus. And also we have other mechanisms like GAPDH is one of the most abundant proteins, made up to 10% of all proteins inside the cell are made of this enzyme. As you see here, it uses NADH. It can be a source for NAD pool here during gluconeogenesis. But also interestingly, if you use glycerol in gluconeogenesis as a source for the fuel to make glucose, then in this case, this guy uses NAD plus. So this one increases NAD plus, but this one uses it. So there's a kind of reciprocal relation between these two. And also, it should be mentioned, I think, checked separately. And maybe we need also to look at the specificities of these two enzymes and many others. But glycolytic pathway is very critical, especially from the viewpoint of cancer. Therefore, we need to check the selectivity of those two genes, enzymes, toward deuterium. So when we look at the proton channels, seems very critical from the viewpoint of the toxicities or other effects of heavy water and deuterium. And studies show that the velocity rate or transfer rate of deuterium from proton channels is 2 times slower compared to hydrogen, and I think it's one of the important things in the field. And also the opening rate of channels and also maybe closing rates are different when we look at hydrogen and deuterium separately. And it will also cause a kind of change in the PDA value up to maybe 0.5 arbitrary units. So one of the systems that is used as a model for proton channels is grammycidin A channels, and it was also shown that conductivity is decreased when we exchange normal water with heavy water. So it causes a kind of 50%. The conductance decreases in the case of heavy water up to 50%. So it says is that, so it kinds of deuterium seems like sticky. So when it goes inside the proton channel, it's kind of sticks there, and it becomes for the channel to expel deuterium when compared with proton. So again, also grammycidin, it also affects the conductance of other ions like sodium and potassium, also are affected by the presence of heavy water. One of the mechanisms that I think that will help us to explain the toxicities or other beneficial or other effects of the deuterium is to look at proton-coupled electron transfer systems. Electron transport system. When we look at the electron transport chain in mitochondria, NADH gives electrons to complex 1, FADH2 to complex 2, and now electrons are accepted by ubiquinone and then transferred to complex 3, and then by cytochrome C to complex 4. But especially complex 1, 3 and 4 use PCET, proton-coupled electron transport system. So whenever an electron passes through them, one proton is sent to the other side of the membrane. So this generates the proton gradient here. So I think that whenever a proton, a deuteron enters the channel, so it will also cause a leakage of electrons from the system. So I think it needs to be studied as one of the main contributors of free radical production as a consequence of normal metabolism, I think. So this is how the system works. So when the electron cannot pass, it goes and binds to oxygen in the vicinity and generates superoxide radical. And this is one of the major sources of free radicals in our cells during normal metabolism. So there are many studies, maybe contradictory studies or results from the studies related to the effects of free radicals, even if they are beneficial or harmful. I think ROS are required at the basal level for beneficial ROS signaling pathways. There are some pathways that require ROS to be activated. Therefore, ROS signaling is very critical, even in the very first stages of caloric restriction. That is one of the most powerful techniques, ways to increase lifespan in different organisms. At the very early stages of caloric restriction, there is an increase in ROS production, and it's very critical for to observe the effects. And electron leakage by detonation in proton-coupled electron transfer reactions can be the major source of endogenous free radicals. And this can explain why some low concentrations, like as mentioned in many studies here today, like 25 ppm of deuterium-depleted was found ineffective, because at these levels maybe it cannot produce sufficient amounts of free radicals or ROS that is required for further signaling. Yeah. So this can explain why some very low concentrations of heavy water cannot be good for some systems, for some specific cancer cells or for some systems like that. So now I'm going to talk about my research on ATP synthase. As you see here, since the dissociation constant of heavy water is lower compared with normal water, so the ratio of deuterons is 1 to 15,000 compared, because it dissociates less, and every second up to 800 protons are passing through this rotor. So it binds here, and then since it neutralizes aspartate here, then the rotor starts to move inside the membrane, inside the lipid. and needs to go through this way, and here it's released to the intermembrane space. So here we have arginine, and in arginine also the binding of deuteron is expected to change the PD up to 0.4. So it's very significant, and binding of deuteron instead of hydrogen here in this system, I think can have a stuttering effect on the turning of this rotor. And when this rotor cannot efficiently turn, it will not generate ATP. So I think that this can be one of the maybe mechanistic explanation for the different types of effects that we observe by the consumption of heavy water. So when you look at the parameters here, the half-life of C subunit is up to 40 to 50 hours, and so in liver mitochondria we have up to 15,000 ATP synthesis. So they have a kind of half-life, but they are continuously maybe reproduced again and again. But anyway, I think that as a kind of steady state level, we could observe a kind of increase in the ratio of deuterated or deuterated ATP synthesis by the passage of time, and this can be one of the major causes for aging, I think. So when we look at the reactome, this is one of the greatest pathways, databases, Japanese pathway, I'm sure you all know it. So when we look at the reactions that use water as an input or output, the total number is more than 4,000, and half of them comes from the ones that use water as a substrate, and the remaining 2,007 comes from the ones that produce water during it. And all of those 4,000 different types of reactions can be scrutinized, I think, very carefully in order to maybe discover some mechanism related to heavy water. So enolase is one of the greatest examples for this one, because it uses during the glycolysis here, it generates water, but during gluconeogenesis it uses water, and one of the most active enzymes in living cells. Especially it's very important for cancer cells, because glycolysis is very active in cancer cells for the production of ATP. So there are great studies concerning, and some of them also mentioned today by other speakers. Amino acid hydrogens can be classified on the proteins as labile, semi-labile, or stable. And when we look at the room temperature, almost all of the labile electrons, labile hydrogens can be changed, protons can be exchanged by deuterium. So this would have some conformational effects, or can change significantly the three-dimensional structure of the proteins. So this is a published list of some amino acids and some peptides, and the exchange is expected as it fits to the numbers that are the calculated ones. And this is for hemoglobin. As you all know, red blood cells live up to 120 days, so there's still too much time for the replenishment or resynthesis. Therefore, almost all of the labile hydrogen parts are maybe exchanged during that period by deuterons. And as you see here, this is true for insulin and other kinds of proteins also, especially the long-living proteins that are found in our lens, in our eyes, or some of the very long-living proteins in our organs, like which are rich of post-mitotic cells, like brains, our muscles, etc. I think are more subject to this kind of accumulation of deuterons during passage of time. And this can be one of the other mechanisms, stochastic mechanisms for aging. So when we look at since the title of my talk is about physiome level changes, so I checked the literature related to especially for some harmful effects of heavy water, very historical articles especially, and the testicles were found to be very sensitive, and it caused, heavy water caused chromosomal damage, and affects heart muscles, and almost every system in our body is affected by heavy water. Since glomerular filtration rate is affected by it, but even though it's very toxic, it's kind of reversible, some of the effects. So when there's a study about sterility, heavy water causes sterility in model animals. Therefore, I recommend that maybe in some unexplained cases of sterility, we can recommend as maybe physicians to our patients to use maybe light water as a kind of alternative approach in addition to their normal routine therapies. There's a study about the increased sensitivity by the addition of heavy water, so it increases sensitivity to radiation. So this is a fish experiment. As you see here, in the presence of heavy water, their lifespan really increases when they are exposed to radiation. And this is lifespan of the effect of heavy water on the lifespan of fish again. As you see here, this is normal water, and this is 25%, and this is 40%. So it really affects in fish systems also. It was shown to affect nerve conduction in frogs. And in cancers also, as is one of the main topics of this conference, it was shown beneficial, or sometimes heavy water in this one. In this case, it's a very old article, but it was shown that here, this is 25%. This is implanted tumors in mice. So they implanted tumors into the peritoneum of the mice. And at this, so this is the controls, lived very short, and the 25% heavy water led them to live longer compared to ones. And this is continued up to 30% of heavy water, but it's neutralized when it's reached up to 40%. So this is one of the classical articles. So heavy water sometimes in some studies seems beneficial. Maybe it depends on the system or cell or whatever, maybe cancer types. Maybe there are some cancer types that's going to be especially sensitive to deuterium or sensitive to maybe low deuterium concentration. As already mentioned in many by other speakers, also deuterium is also one of the important topics nowadays in the pharmaceutical industry because of the CD bond is more hard to break compared to CH bond. And also this is already GMAC's article, and it's deuterium-depleted water, so compared with heavy water, is effective even in behavior or in even microRNA profiles. So it has good or beneficial effects. So when we get to, so my suggestion for some questions, and I'd like to discuss with you, and for some future prospects, I think that one of the questions that we need to answer is, are there some regulatory or structural macromolecules, they can be enzymes or other macromolecules, that selectively prefer and use deuterium for their functions? So instead of accepting it as a kind of on the isotope of heavy water, I'm sure that since we are all our evolutionary system, evolution during whole evolution period, so deuterium was there. Therefore, it has very critical change differences compared with hydrogen. Therefore, we would expect that there are some specific enzymes or other macromolecules that specifically use and prefer deuterium. So we need to discover them. So what kind of strategy can be to scrutinize, maybe check, make a kind of a screening study to check in which enzymes the activity increases in the presence of deuterium. So another one, as I mentioned at the beginning of my talk, can deuterium be pragmatically accepted as an element, not only as an isotope of hydrogen, instead of trying to explain its biological effects only in the context of kinetic isotope effects? So another one is, does deuterium cause electron leakage in proton count electron transfer system? I think This is one of the questions that we need to answer. So if yes, it provides kind of, I think, sensor for us. So that responds to very loud changes in this concentration of deuterium in the electron transport chain, with chain's vicinity. And also we need to synchronize all chemical reactions that use or produce water. And also one of the most critical ones, I think, is to start a randomized, placebo-controlled, double-blind, very high-quality prospective clinical study, maybe by using twins, to test the effectiveness of deuterium-depleted water concerning its effects on metabolic parameters or aging or well-being or whatever. So I think this is a must in order to convince other people for the effectiveness of the light water. And also we can use imaging studies like MRI to check if the deuterium accumulates in organs, in our tissues when we age. So it would be especially in brain or other muscle type organs that are rich of post-mitotic cells. So actually we did this kind of study. So we checked the concentrations of if there is an accumulation by the passage of time in the tissues of mice. So these are the individual values for each mouse. So when we compared 4 and 5 weeks, there seems kind of an increase. But we lost, or even we saw decreased levels of mice after 1 year. So we have no data about how to explain this finding. Maybe among you, some of you can help us concerning this one. Another one that I would like to mention is lysosomal pH increase. So as maybe some of you know, one of the first signs of cellular senescence is an increase in the lysosomal pH. And lysosomal pH seems very critical. It's a kind of hub for many types of age-related diseases and pathways, etc. And the tradition of V8 can have an effect on lysosomal pH, I think, because it works like 80% works, as you see here. So it has almost the same structure, and it also it has something especially significant in, I'm going to show here, I'm sorry, in cancer cells. You know, cancer cells are somehow acidic, and when V8 the cell cytoplasm is acidic, then V8 relocalizes to the cell membrane and starts to export hydrogen ions. Therefore, it's very critical for tumor cells, and maybe some of the toxic effects of heavy water in tumor cells can be explained by the stuttering of V8pases. If they are not able to export or excrete hydrogen, so maybe the levels of pH levels will not be able them to survive anymore. As you see here, mitochondrial ATP synthase and V8Ps are very similar in structure, and even though I didn't finish my calculations yet, so the same effect can be expected here. So entrance of deuterium to the rotor part of the V8Ps can also make it stutter. So in humans, the system works only in one direction, but in there are some special organisms that use the same motor to produce ATP also. So if the protons goes in the reverse direction, it can help the system to produce ATP. There are some organisms that use the reverse type of this reaction. Maybe it can be a way to maybe compensate by genetic engineering approaches or something in the future, to use that system in human cells also to generate ATP by using lysosomes. So it would be an interesting approach, I think, in the future. So this is how I explained the stuttering of ATPase. So these are my final works: the slowing down of biochemical reactions and kinetic isotope effects that occur by the replacement of hydrogen that might be contributing to stochastic mechanisms of aging and age-related disease at a physiome level, I think. And also we need a kind of physiome level studies and maybe data mining analysis of all metabolic pathways, cellular processes and biomolecules that are sensitive to replacement of proteome with deuterium and possibly involved in aging needs to be carried out by the researchers of the field. And now I'm going to finish my talk. Thanks to my family, because I use their time in my studies, and especially to thank Viktor Sagalowski for kindly inviting me here, and otherwise I wouldn't be here, and also organizing committee for kindly helping me. And this is, I'd like to finish with Mardin. I don't know if you know this city. It's one of the ancient, most beautiful cities in Turkey, historical cities, and there is we call it Kasimiye, but it's a kind of ancient university, ancient school. It's called Kasimiye, and here there's a fountain and pool, and it represents the whole life of a human being. So here, at the beginning of life, so everything is kind of mess, and then we have some calm times, and then we go. So it's related to water. Also, I wanted to put it here. Okay, thank you very much for kindly listening to me.